Assalamualaikum and hi. In this video, we will be presenting about tissue sectioning in histology. This video was prepared by me, Ahmad Nabil bin Nur Rahizan, and then Amma Iman bin Nur Azli, following by Amila Jibrin, and lastly, Azrul Muhaizi binti Nur Azhari. First thing first, in this video, I will introduce you about the tissue sectioning. So what is tissue sectioning? Okay, tissue sectioning is a key technique in histology that allowing us to visualize the microstructure of tissues and a cell under a microscope. This process is very essential for identifying cellular details and understanding tissue organization, which are critical for diagnosing disease, studying biological process, and advancing medical research. So, what is tissue sectioning? Okay, tissue sectioning is the process of slicing very thin pieces of tissue for examination under a microscope. This technique is crucial in histology and pathology because it allowing scientists, doctor, medical officer, or researcher, or even student to study the structure and detail of cell and tissue in various organs. Next is purpose of tissue sectioning. So what is the purpose of tissue sectioning? So the purpose of tissue sectioning is to obtain clear image of tissue and detailed image of tissue and cellular structure for observation under a microscope. This is very essential in diagnosing disease, researching cell biology, and understanding tissue organization and pathology. Tissue sectioning is a fundamental technique in histology and pathology that allows researcher and medical professional to observe the microscopic structure of tissue and cell. By cutting thin slice from a tissue sample, sectioning enable detailed examination of cellular morphology, tissue organization, and potential abnormalities, which are very essential for diagnosing disease and conducting research. The process involves several key steps, which are the typical sectioning workflow. The first step is fixation that preserve tissue integrity by preventing decay. And the next step, we have embedding that support the tissue, usually with paraffin wax or freezing, to allow precise cutting. And the next step, the third step is sectioning. We use a microtome to slice ultra thin section, generally between 3 to 10 micron thick, so light can pass through for imaging. And then the next step is staining. It is to highlight specific cellular structures using dyes like hematoxylin and eosin to enhance contrast. And lastly, we have mounting to secure the section on a glass slide with a cover slip for a long-term study.
For the next part, we would like to talk about advancement of the sectioning process. The advancement for the sectioning process is called a vibratome. A vibratome is a scientific equipment that is used to cut small tissue slices for microscopic inspections. It functions similarly to a microtome, but it cuts tissue with a vibrating blade rather than a stationary one. By reducing pressure on the tissue, the vibrating blade helps lessen compression artifacts. How does a vibratome function? Number one, the tissue sample is mounted securely on a platform or specimen holder. This will keep the sample balanced during cutting. Second, the blade then vibrates at a controlled speed of frequency and amplitude. The vibrating blade decreases the friction and force applied to the sample, allowing smoother cuts which can preserve the tissue's delicate structure. Lastly, after the thin section is produced, these samples are collected in a buffer or on a slide to prevent damage. You can see an example of the procedure and preparation of a mouse brain for vibratome sectioning. We'll be talking about the comparison of the advancement method versus the conventional method. The machines are the microtome and vibratome. The type of blade used for the microtome is a stationary blade, while the vibratome uses a vibrating blade. The thin sectioning of the microtome is 1 micrometer to 10 micrometer, while the vibratome uses a thick sectioning with 30 micrometer to 40 micrometer. The type of sample used are also different. The microtome uses a paraffin embedded tissue, while the vibratome can use soft and live tissue. Part is vibratome's advantages and disadvantage. For the advantages, in contrast to microtome and cryostat sectioning, where the blades are static, the oscillating knife results in less section compression and result fewer section artifacts. And a clearer three-dimensional view of the sample structure is made possible by thicker sections. While the disadvantages is due to the thicker sectioning result, it will be hard to analyze the tissue on a light microscope. To overcome this challenge, an electron microscope would be easier to use.
Traditional and conventional methods are used in histology laboratories. In histology, traditional and conventional are terms frequently used interchangeably, though they can have subtle differences depending on the context. Broadly speaking, both refer to standard, routinely practiced methods for tissue preparation and sectioning. However, making a distinction between the two can reveal some nuances in histology techniques. So, these are the differences to both methods, as you can see here, traditional sectioning and conventional sectioning. For automation, traditional sectioning are manual, little or no automation, while conventional sectioning uses automated or semi-automated equipment. For technician skills, traditional methods are high skill required, and conventional methods still require skill but less manual control. For time efficiency, traditional methods are more time-consuming than conventional methods that are faster and more consistent. For consistency, traditional methods may vary depending on technician skill, and conventional methods generally more uniform and reproducible. For tools used, traditional methods are basic microtomes, often hand-driven, while conventional are automated microtomes, tissue processors. For application settings, traditional sectioning have low resource labs, smaller settings, while conventional sectioning are modern, well-equipped labs. For fixatives and embedding, traditional methods are often limited to formalin and paraffin, while conventional methods can include cryo methods and other fixatives. Although both traditional and conventional sectioning methods strive to produce high-quality tissue sections, conventional techniques integrate more technology and automation, enhancing efficiency, consistency, and throughput. Traditional methods, which are mostly manual, remain common in smaller labs, where access to advanced equipment may be limited. Conventional tissue sectioning is a critical step in histology, requiring the slicing of thin sections from embedded tissue for microscopic analysis. This process presents several challenges due to the complex structures and fragile nature of biological samples. Here are some of the main challenges encountered in conventional tissue sectioning. Risk of artifacts. Artifacts like faults, wrinkles, and compression can develop during sectioning, distorting the tissue sections. Such artifacts may obscure key details or result in diagnostic misinterpretations. Time and labor intensive. Manual sectioning can be slow, particularly with large tissues or when multiple sections are needed. Achieving high-quality sections often takes several tries, adding to time and labor cost. Temperature sensitivity. Wrong temperatures during sectioning can harm tissue quality. Overly warm paraffin-embedded tissues may soften and compress, while overly cold ones can shatter. Cryosectioning also has similar temperature issues. Thickness inconsistency. Keeping sections consistently thick, usually 3 to 5 microns, is important for even staining and clear analysis. Manual sectioning can create uneven thickness, leading to staining issues and making it harder to compare sections accurately. Blade wear and maintenance. Microtome blades gel quickly, especially when cutting fibrous or calcified tissues, which raises replacement costs. Dull blade also heighten the risk of artifacts and tissue damage. Conventional tissue sectioning is managed levels, like keeping sections even, dealing with artifacts, and handling different tissues. While new technology like automated microtomes and cryostats help, skilled technicians are still needed for the process.
Hi, Assalamualaikum. My name is Azrul Mohisi binti Azhari, and I will continue with the last part, which is the conclusion. To sum up, the conventional method will producing reliable and superior slides. Bringing new technology into histology workflow can enhance the diagnostic accuracy and speed. Traditional method call for expert manual manipulation to maintain section integrity, but implementing advancement technology like vibrotome can improve the histology workflow, lower human error, and easier. The balance of conventional method and new technology will boost both superior results and the changing needs of medical research and diagnosis. So this is our references. And that's all from us. Thank you very much for watching.